Have you ever stopped to think that inside you, right this very second, there's a microscopic drama playing out millions of times over? It's this incredibly precise, choreographed performance that lets you grow, heal, and, well, exist. Today, we're going to pull back the curtain on this amazing cellular ballet, mitosis. So grab a seat because the show is about to start. So the star of our show is what's called the M phase. Our source material calls it the most dramatic period of the cell cycle, and for good reason. This is where one parent cell splits into two identical daughters. It's also known as equational division. Now that's a really key term you got to remember. It just means the number of chromosomes in the new cells is exactly the same as in the original parent cell. We're talking a perfect copy, every single time.
To make sense of this whole thing, scientists have broken down this continuous process into four main acts. We're going to walk through each one: prophase, metaphase, anaphase, and telophase. Think of this as your program for the show, so you can follow along with all the action.
And so our performance begins. Welcome to Act 1, prophase. This is where the cell gets everything ready, sets the stage for the massive division that's about to happen.
This slide gives us a fantastic look at what's going on inside. See, in early prophase, all that genetic material, the chromatin, is kind of messy, like a tangled ball of yarn. But by late prophase, look at that. It starts to condense into these thick, distinct structures. Those are the chromosomes becoming visible. And notice those little star-like things moving apart? That's the machinery that's going to pull the whole cell apart. Okay, so let's break it down. That loose genetic stuff tightens up into these compact chromosomes. And if you look closely, each chromosome is actually made of two identical sister chromatids, and they're joined right in the middle at a spot called the centromere. At the same time, the cell starts building this internal rope and pulley system. Two anchor points, the centrosomes, head to opposite ends of the cell. They start shooting out these tiny ropes, called microtubules, which form those star-shaped things called asters. The whole setup, the anchors, and the ropes is called the mitotic apparatus.
And now, let's move on to Act 2, the great alignment. This stage is called metaphase, and it is all about precision and order. All that prep work from prophase, it's about to pay off right here.
And would you look at that? This picture perfectly shows the defining moment of metaphase. Check out that perfect lineup. The chromosomes are all pulled right to the very center of the cell, forming a perfectly straight line, kind of like they're lining up for inspection at the cell's equator.
So how in the world does the cell pull off this perfect lineup? Well, first, the container holding all the chromosomes, the nuclear envelope, it just dissolves. Now those ropes, or spindle fibers, can finally grab on. They attach to these special little handles on each chromosome called a kinetochore. And then, acting like a bunch of tiny tugboats, these fibers pull and push the chromosomes around until they are perfectly aligned along this imaginary line in the middle. We call that line the metaphase plate.
The stage is set. The actors are in position. And now, for the moment this entire process has been building towards.
We've reached the climax of our performance, folks. Act 3, anaphase. This is the shortest, but without a doubt the most action-packed stage of mitosis.
Just look at the motion here. The sister chromatids, which were perfectly paired up just a second ago, are now being yanked apart, moving to opposite ends of the cell. You can even see the cool V-shape they make as they get dragged through the cytoplasm. It's happening.
The action is so fast and perfectly synchronized. At the start of anaphase, every single centromere splits at the exact same time. Pop! This is the moment the sister chromatids finally separate. And once they're apart, we don't call them chromatids anymore. Each one is now a full-fledged daughter chromosome. The spindle fibers get shorter, pulling these new chromosomes to opposite sides, with the centromere leading the charge and the arms of the chromosome just trailing behind.
Okay, after that intense climax, it's time for the resolution. We are now entering the final act, telophase, where the cell puts the finishing touches on its division and gets ready to become two separate cells. This is followed by the final split, a process called cytokinesis.
And as you can see right here, telophase is pretty much prophase but in reverse. The chromosomes have made it to their destinations at opposite ends of the cell. Now the cell starts to rebuild all the stuff it broke down earlier, forming two new complete nuclei.
So what's happening in detail? Well, the chromosomes, now at the poles, they start to uncoil and decondense. They lose that distinct shape and turn back into that messy ball of yarn, the chromatin. A brand new nuclear envelope forms around each of these two clusters of genetic material. And finally, other organelles like the nucleolus, the Golgi complex, and the ER, they all reappear. The cell now officially has two fully working nuclei.
But wait, the job isn't quite done.
The nucleus has divided, sure, but we still have one giant cell with two command centers. The final step is to actually split the cell itself. This is cytokinesis, the physical division of the cell's cytoplasm to create two totally separate daughter cells.
Now, what's really cool is how this happens is different in animal and plant cells. In animal cells, like ours, a furrow appears. It's like pulling a drawstring on a bag. It just pinches inward and inward until the cell's divided in two. But plant cells, they have a tough, rigid cell wall, so they can't just pinch. Instead, they build a new wall called a cell plate right down the middle. It grows outward until it connects with the existing walls, creating a brand new boundary between the two daughter cells.
So, we've seen the whole performance, from start to finish. But what's the point of this whole elaborate cellular dance? Let's talk about the encore, the significance of mitosis. Why does this matter so much?
Well, first up, growth. Every single one of us started as a single cell, a zygote. Mitosis is the process that turned that one cell into the trillions that make you. Second, repair. When you get a cut, or even just through everyday life, your body uses mitosis to replace old or damaged cells, like in your skin or the lining of your gut. It also makes sure cells don't get too big for their own good, keeping everything running efficiently. And maybe most importantly, because it's an equational division, it makes sure that every single cell in your body has the exact same genetic blueprint. So the grand performance of mitosis is this masterpiece of biological precision, designed to create perfect copies to build and maintain us. It's the reason we are who we are, literally, cell by cell. But that leaves us with a really important question to think about. This process is built for perfection. So what happens when it makes a mistake? What happens when the copy isn't perfect? Well, that's a story for another day.